This next section of the Unit 3 lecture covers a lot of information, so I'm going to break this last section up into two lecture videos. So the first one, we're going to be talking about getting into this group of organisms known as the Enterobacteriaceae. So this first video, I will do just kind of the intro and the general characteristics of that group of organisms, and then the testing that's associated with them. And then the second part to this section of the lecture will be we'll get into the individual organisms themselves. Okay, so general characteristics, and again, this is this group of organisms in the family Enterobacteriaceae. So these are characteristics that would be found in all or most of these organisms. So family is Enterobacteriaceae. This includes 44 genera, 176 species, and they're often referred to as just kind of collectively as the enterics, meaning that they are of or related to the digestive tract. They are all either gram-negative rods or coccobacilli. They are motile at body temperature, and there's a couple of exceptions to that. So Klebsiella, Shigella, and Yersinia are not motile. If they are motile, then they're paratrichous, which, if you remember from Unit 1, means that they have flagella all the way around. They are all glucose fermenters. They are all oxidase negative, meaning that they do not produce the enzyme cytochrome oxidase. The exception to that characteristic is the organism Plesiomonas, which, that Plesiomonas, the organism recently was added to this group of enterics based on DNA studies, but it kind of has a lot of characteristics of a different group that we'll cover later. And then they are all considered antigenic. So antigenic substances are those that can stimulate production of antibodies. So if our body comes in contact with these organisms, then our body has an immune response to that and forms antibodies. They are all rapid growers, and when we're talking about microbiology, that's kind of relative. But so the Enterobacteriaceae, when we say rapid, you'll usually see colony formation at around 12 hours of incubation. They are ubiquitous in nature, so some species exist as free-living organisms in water, soil, or sewage. Some are plant pathogens. Additionally, the Enterobacteriaceae, with very few exceptions, share a common characteristic. They reside in the GI tract of humans. So the exceptions to that would be Salmonella, Shigella, and Yersinia. Their colony morphology on your blood agar plate is of little value because most of the enterics look the same. They're going to be large, shiny, wet, gray colonies. Klebsiella, Proteus, and Enterobacter are three organisms that have more distinct colony morphology, and we'll talk about that when we get to those organisms. They are all facultative anaerobes, which means that they can live with or without oxygen, but they do grow better in aerobic conditions, but they're able to grow in anaerobic conditions also. They reduce nitrate to nitrite, and again, a couple of exceptions here. Photorhabdus and Xenorhabdus are not capable of doing that. They are non-spore formers, and they're all commonly isolated Enterobacteriaceae are catalase positive, except for Shigella dysenteriae type 1. So again, this would be a reason why the gram stain is so important, because if we have an organism on the plate that looks like, it might look like a staph species. Remember back to Unit 1, our staph species are catalase positive, so this can lead us down the wrong testing rabbit hole if we're not careful. Okay, some virulence and antigenic factors. So virulence of the enterics is affected by a lot of factors. So one of those things is their ability to colonize, adhere, produce toxins, and invade tissues. Some of these organisms contain plasmids, which may help mediate resistance to antibiotics. So plasmid is just kind of like a little extra piece of DNA that some bacteria have that carry virulence genes and antibiotic resistance genes. antigens. So a lot of the enterics possess antigens that can be used to serologically put them into groups. So that helps us in testing and identifying these organisms. One of those is the O antigen, also known as the somatic antigen, which is located in the cell wall of the organism. It is heat stable, which means that heat will not affect it. You've also got the H antigen, which is a flagellar antigen, so it's an antigen that's found on the surface of flagella. It is heat labile, which means that it's susceptible to the effects of heat. And so if an organism possesses the H antigen, that means that the organism is going to also be motile. And then the K antigen, which is also referred to as the capsular antigen, it is heat labile also. It's a polysaccharide found only in certain encapsulated species, and it will mask the O antigen, the heat stable cell antigen. So if we were testing an organism for these different antigens, if the organism had the O antigen, but it also had the K antigen, and the K antigen is capable of masking the O antigen, then how could we determine that the organism, that O antigen was present? Well, the O antigen is heat stable. The K antigen is heat labile. So you could heat the specimen. That would get rid of the K antigen, and then we would be able to detect the O antigen. Okay, preliminary identification. So this is, we're going to go through the different media, which some of this we've already been through, and different testing methods that could help us identify these organisms. So first is our differential and selective media, which we've talked about since, I think, the very first lecture. So these organisms utilize sugars in the media that cause color changes, which show the organism's ability to ferment the sugar. So this helps with our identification of these enteric organisms. Okay, McConkey, again, and your McConkey and your EMB are kind of interchangeable, so your lab would only use one of those. The McConkey is, and both of those are selective for gram-negative rods, which means they've got stuff in the media that suppresses the growth of your gram-positive organisms. So we know that if an organism is growing on the McConkey or the EMB, it has to be a gram-negative rod. But then these media are also differential based on lactose fermentation. So on the McConkey and the EMB, which both start out kind of a pink color, once those organisms ferment the lactose, you're going to see a color change in the media, which is what makes these media differential. So on your McConkey, lactose fermenters are going to appear as dark pink colonies. Non-lactose fermenters will not change the color of the original media, so the colonies kind of stay clear to pink. They kind of blend into the media. Same down here with the EMB, except you're going to have a darker purple or a dark pinkish purple with those lactose fermenters. But same, no color change with the non-lactose fermenters. Hectoin, if you remember, is media that's used for isolation of Salmonella or Shigella. It also has lactose in it, so it differentiates based on lactose fermentation, but it also differentiates based on hydrogen sulfide production. So your lactose fermenters will change the color of the media from the original kind of green to this orange yellow. Non-lactose fermenters will not change the color of the media, so they will remain that original color of the media, which is kind of a blue-green. And then organisms that are capable of producing hydrogen sulfide, you'll see black colonies or black dots in the center of the colony. And then again, on your blood agar plate, kind of nondescript colonies, most of them are going to look the same. And here's, I'll let you guys look through this chart, but this just kind of breaks down the different media types that we have talked about. And all of these are selective and differential media. So it has on here the thing that has been added to the media that makes it differential. what makes it selective. So this is what has been added to the media that allows only certain organisms or types of organisms to grow, those that have the hydrogen sulfide indicator in them, and then what the different colors will, organisms will appear as based on whether or not they've utilized the lactose or not. We've seen a couple images of hectowin so far. So this one has got a lactose fermenter streaked on it here, and they've used Klebsiella pneumoniae, and you can see it has utilized the lactose. It has changed the color of the media from this green to this orange, and then over here, I'm not sure what organism that is, but it is a non-lactose fermenter, and it has not utilized the lactose, so it has remained the original color of the media. Same here, that's just a close-up of another organism growing on hectowin that has not utilized the lactose. And we've seen this same picture a few times, I believe, but we've got hectowin with, you've got a lactose fermenter, which is up here, the orange. We've got hydrogen sulfide production that we can see there as indicated by that black colony. And then if you look at some of these smaller colonies here outside of the black dot, you can see the edges of the colony that are kind of the green, so they're non-lactose fermenters. We've looked at this same picture with the EMB previously also. So we've got 3 organisms growing on this plate. The media is selective, so we know if we've got growth that the organism has to be a gram-negative rod. On the left over here, we've got a non-lactose fermenter. So you can see the colony or the organism streaked on there, but it has not changed the color of the media because it did not utilize the lactose. Whereas in the center here, we've got an organism that has utilized the lactose, so it's changed to just kind of this darker purple pink color. And then remember, E. coli is the one that's kind of weird. He's the only one that will show up this way with that green metallic sheen. Okay, now we're going to be getting into some of the testing that's used to differentiate your Enterobacteriaceae. So oxidation fermentation tests. So this is a type of carbohydrate fermentation test. It's a solid medium, can help in the identification of bacteria, particularly in the differentiation between the enterics, which ferment glucose from other similar gram-negative rods. So it determines the ability of the organism to ferment specific carbohydrates. So some organisms ferment a single carbohydrate while others ferment or oxidize several carbohydrates. So fermentation is an anaerobic process where organic compounds are converted to similar compounds. The end product is acid or acid with gas, which is detected by a pH indicator that's added to the medium, and more acid is produced during the process of fermentation versus oxidation. So oxidation is an aerobic process of producing energy, so it further metabolizes the acid that's formed from fermentation into CO2. The process of oxidation requires oxygen since it's an aerobic process. So the testing that's performed, so you've got 2 oxidation fermentation medium tubes that get inoculated. The fermentation tube, and let me take you to a picture here so maybe this will make more sense as I'm talking about it. The fermentation tube is overlaid with mineral oil. I think they also use paraffin sometimes also to produce that anaerobic environment. The oxidation tube is left aerobic. So here's just the difference in like a tube that has, that's what that's supposed to represent there, is the mineral oil that's laid over top. So this would be your anaerobic tube. This would be your aerobic tube. The uninoculated tube is green. If the organism produces acid, it will turn yellow. So how can we interpret these different results? So if acid, let me back up for just a second. So again, the media uninoculated is green. Your pH indicator that is in this media is called bromthiamyl blue. A positive reaction, which means that acid was produced, causes a yellow color change in the media. Negative result, which means the environment has remained alkaline. So you might have growth. The organism may have been able to grow in the media, but there's no color change. And then if there's no growth at all, this is called the organism would be referred to as a non-utilizer. So the organism is not able to grow in the media at all. So the interpretation of these different results. So if you've got acid produced in both tubes, so that would be here because we've had a yellow color change in both our aerobic and our anaerobic tube, then the organism is an oxidizer and a fermenter. If acid is only produced in the closed tube, so the closed or the anaerobic tube here, sorry, I'm on the wrong one. Acid is only produced in the closed tube, then the organism is a fermenter and possible obligate anaerobe. So that's the tube where the mineral oil was overlaid. So this produces the anaerobic environment. So if we've got color change in this tube, in your anaerobic tube only, then that means that your organism is a fermenter and possibly it can only grow in an anaerobic environment. If we have acid only in the open tube, so that would be this one, where we've had the yellow color change in the aerobic tube, but not in the anaerobic tube, then that organism is an oxidizer and a non-fermenter. So no acid production in either tube means that the organism is a non-oxidizer or possibly non-saccharolytic. So a lot of non-oxidizers will produce alkaline reactions from peptone utilization, so you might actually, it could turn the media red. Your control organisms for this test, your glucose fermenter is E. coli, glucose oxidizer is Pseudomonas aeruginosa, and non-saccharolytic, which means that it is not utilizing the carbohydrates, is Moraxella. And if you remember from Unit 2, that was how we differentiated Moraxella from the Neisseria species, was that Moraxella did not utilize carbohydrates. Okay, the next test is called the TSI, or triple sugar iron auger. So this is another type of carbohydrate utilization test. It determines the organism's carbohydrate utilization, and the sugars that are incorporated into this test are glucose, lactose, and sucrose. This test also has the ability to detect gas production and production of hydrogen sulfide. So it's primarily used to differentiate the Enterobacteriaceae from other gram-negative rods. So this one is also a slant auger, and just briefly I'll show you that picture so you'll know what I'm talking about. So it's a slant auger made up of 0.1% glucose, 1% lactose, and 1% sucrose. And that is, so in these slant medias, this area right here, which is the slanted portion of the auger, is referred to as the slant. This portion down here, which is the anaerobic portion and the bottom of the tube, is referred to as the butt of the tube. So that sugar that's added to the media, that 0.1% glucose is present in the butt. The lactose and the sucrose is present in the slant. So once we look at the results of this test, that's what helps us determine which sugar has been utilized. The pH indicator in this one is phenol red, so it will turn yellow at a pH below 6.8. So that means acid has been produced in response to utilization of the sugar. There's also ferrous sulfate, which helps us detect production of hydrogen sulfide gas. And then there's also a 0.1% concentration of peptone in the media, which is a protein. So the uninoculated medium is red, And again, the slant portion is aerobic. The deep portion, which again is referred to as the butt, is anaerobic. Okay, interpretation of these results. So these are ones that you're going to have to inoculate the media with the suspected organism. It has to incubate, and then we're going to read the results between 18 to 24 hours. The results are written as your slant result over the butt result. So I think I've got an example. I'll show you of that in just a minute. So we use a K to indicate an alkaline reaction. And again, alkaline means that acid has not been produced, so the tube has stayed the original red color, so that's considered negative, and no fermentation has taken place. We use the letter A to indicate acid production, which is considered a positive reaction. The media has fermented the sugar, produced acid, and turned yellow. We use a plus sign to indicate hydrogen sulfide production, and that is indicated by the media turning black. We use a G to indicate gas production, which you'll see in the media as bubbles or air pockets. And then if the organism utilized peptone, the media will change to a deep red or purple. Okay, so again, I said we report these results out as the slant result over the butt result. So this would be A indicates acid. This is the slant. A indicates acid. That's the butt result. So we say it as A over A. And then we would add on that plus sign or the G if there was also hydrogen sulfide production or gas production. If the results, so I said that you have to read the results between 18 to 24 hours. If those results are read too early, a tube that should appear as A over A would appear as K over A. If it's read too late, one that should appear as K over A would appear as K over K. And that's since glucose is present in the media in a lesser amount. If the glucose is utilized, it initially turns the butt and the slant yellow due to that acid production, but as all of that glucose is completely used up, the slant's going to start to revert back to alkaline conditions unless sucrose and or lactose are also utilized. So and if that's the case, then the entire agar would remain yellow. With that, if you read it too early, this is going to happen where you have one that should be A over A is going to appear as K over A, and that's just because it's not had enough time for that carbohydrate to have been utilized yet. And then the KIA is another similar test, one that's similar to the TSI, but only glucose and lactose are utilized and not sucrose. And here's that picture again, just of some different TSI results. So this one, we've got a yellow slant, a yellow butt, which means that the sugars were utilized all the way throughout. So we write that as A over A. In the second tube here, the slant has not changed to yellow. It has remained that original red color. So we report that as K over A. In your third tube, the butt is black and the slant is red. We don't see any bubbles or cracks in the media. So this tells us that hydrogen sulfide has been produced. Glucose only has been utilized. And for hydrogen sulfide to be utilized, it has to have an acidic environment to do that. So you can assume acid in the presence of hydrogen sulfide. So if you're seeing black on the bottom here, you can also assume acid production and no gas production. And then in that fourth tube, it's entirely red, no cracks or bubbles, or alkaline. So this tube, it could be one of two things. That either this tube has not been inoculated, or it could just be that it has not utilized any of the sugars. It's not produced gas. It's not produced hydrogen sulfide. But it says here on the bottom, I can see where it says Pseudomonas. So this one has been inoculated, but it's with an organism that is not in the group of Enterobacteriaceae. This organism is one that is in kind of a larger group known as non-fermenters. So this makes sense that it has not utilized the sugars. Okay, the next test that we're going to talk about is called the MVIC test. And this is actually kind of a group of tests that are performed together, and each one of the letters here stands for one of the different tests in this group. So the I stands for indole, the M stands for methyl red, the V stands for Voges-Proskauer, and the C is for citrate. So indole is a test that's used to identify organisms that produce the enzyme tryptophanase, which enables it to hydrolyze tryptophan to form the compound indole. So this is a broth. It should be inoculated for 48 hours. And then we have to add another reagent. And you can use either Kovacs or Ehrlich's reagent. Positive reaction is going to be a pink to red color. Negative is no color change. They also have rapid indole tests available that are kind of like it's similar to the oxidase test where it's done on filter paper. Okay, methyl red detects the end products of glucose fermentation. So bacteria get incubated in the broth medium that contain glucose. That broth is incubated for 3 to 5 days. After that 3 to 5 day incubation period, half of that broth gets transferred to a clean, dry tube for the Voges-Proskauer test. If glucose is metabolized, then a bright red color develops after the addition of the pH indicator, which is methyl red. And then methyl red negative cultures remain yellow. Okay, your Voges-Proskauer, this also detects end products of glucose fermentation. Acetone, which is also known as acetic acid, is oxidized to a diacetyl after the addition of alpha naphthol and KOH. So this is one that requires both of those reagents. So you have to add alpha naphthol and KOH. Excuse me. Red is a positive color and yellow is negative. Side note, bacteria are usually going to be positive either for the methyl red or the Voges-Proskauer. Rarely will they be positive for both, but they could be negative for both. So enterics can use different pathways to metabolize glucose. So your methyl red test and your Voges-Proskauer test are testing for those two different pathways. Okay, and then the last test included in the MVIC is the citrate test, which is used to identify organisms that use sodium citrate as their only carbon source and inorganic ammonium salts as their only nitrogen source. So Simmons citrate media is the most commonly used. It is a slant auger. Positive reaction results in a green positive reaction results in a brilliant blue. The uninoculated or negative result is green, so it starts out green. And here's just a couple of pictures of these different tests. So your indole test, negative, and positive is changed to that pretty kind of magenta color. Methyl red, so we've got one that's not been inoculated here, one that has remained negative. So you can see it's turned cloudy. So that indicates that the organism has grown in the media, but it's not a positive result. Over here's positive. A couple different variations on results with the Voges-Proskauer test. And then your citrate test, which here is uninoculated or negative. And on the left is the positive, bright, pretty blue color. Okay, the next test is the LIA. LIA stands for lysine iron auger slant. So this is a solid slant auger also. It's detecting decarboxylation or deamination of lysine. So decarboxylation is an anaerobic process. Deamination is an aerobic process. This one also has the capability to detect hydrogen sulfide production. Your pH indicator in this media is bromcresol purple, and your uninoculated tube is a light purple. Black precipitate in the butt of the tube indicates hydrogen sulfide gas. And then purple, if you see a purple result, this is positive for decarboxylation of lysine, so that would be indicated as a purple result in the butt of the tube. A red or burgundy, which would be indicated in the slant, is positive for deamination of lysine. And then yellow is, in either the slant or the butt or both, is a negative reaction. So we've got a couple of different results here. So your C is the control tube. Number 1 is negative for deamination of lysine, negative for decarboxylation. Let's see, there's one of these I was going to, I don't see the one I'm looking for, but so just look at the various reactions here and relate those to the descriptions in the notes section, and that just kind of shows you some of the various reactions you can get from your LIA. Okay, the next test is urease. This one again is a solid slant auger, determines hydrolyzation of urea by detecting production of ammonia. Urease hydrolyzes urea to form ammonia, which produces this color change with the pH indicator. Christensen's media is generally the preferred media for urease detection. So the slant is inoculated, but it's not stabbed. We have to incubate. We read the results after 24 hours. A longer time, so if we wait too long to read those results, it can give a false negative. For this one, your pH indicator is phenol red. Your positive result turns a bright magenta color, and a negative result is kind of an orangish color. And here are a couple different examples of those urease reactions. So this one is considered rapid positive. So the reaction occurs so quickly that just that top portion turns. This one is a negative, and then this one is just your traditional positive. Okay, the next test is SIM media, which SIM, S-I-M, stands for sulfide, indole, and motility. This one is another slant auger where you're going to stab straight into the media, so not at a slant. And this is, you would do this with an inoculating needle versus an inoculating loop. Incubate, read your reaction at 24 hours. So let me just click over here so you can see some of these reactions. This one has an indicator for the hydrogen sulfide production, so that results in a black precipitate. Indole positive is pink or red after the addition of your reagent, which is your Kovacs reagent. And then motility positive is indicated by cloudiness around the inoculation line. And what it means here, like for instance in this one right here, so you can see like in B where they've used the inoculating needle to stab straight down into the media, and there's growth there, but it's not spread out, so it's not moved, it's not modal, whereas in this one you can see how it's kind of spread out here. And same as this one, you can see the hydrogen sulfide production, but you can see how the stab line has spread out. So a few different reactions there. And here's another one just of just motility media to see that difference in, you've got the stab line here, and the organism is capable of growth, but it's not modal, whereas this one has spread out from the center of that stab line. Okay, the next test is the nitrate test. So this is a broth. It determines reduction of nitrate to nitrite, and it can also detect further reduction of nitrite to nitrogen gas. So this one is a little bit tricky. The reagents are added, so your reagents are alpha-naphthalamine and sulfonilic acid after 24 hours of incubation. Okay, your uninoculated medium will be clear. Red or pink indicates a positive reaction, which indicates the presence of nitrite. So that means the bacteria were able to reduce nitrate to nitrite. However, no color change. So if we get a red result, that's positive, we're done. But no color change could mean either that nitrate has not been reduced by the bacteria or that it has further reduced to gas, which those reagents would not detect. So at that point, you then have to add a small amount of zinc dust to determine if you've got a true negative or if the bacteria further reduced to nitrogen gas. So if we have a red result after the addition of the zinc dust, this confirms a true negative, because that means that there was still nitrate left in the media that the bacteria had not reduced, but the zinc dust did. If the nitrate had already been reduced to nitrite, then the addition of the zinc dust would not have had anything to reduce. So if there's still no change after the addition of the zinc dust, you can call that positive complete, and that means that the it had been reduced further to nitrogen gas. So here's kind of some interpretation of those different results. So you've got your original tube. So A is positive. So that means the bacteria reduced nitrate to nitrite. You've got a red result. You're done. However, if you add your reagents and you've got a clear, or it's not changed color, then you don't know which is the case. Did the bacteria not reduce, or did it reduce further to nitrogen gas? So then we have to add the zinc dust. So our result after the addition of the zinc dust is no color, which is considered a positive, because that means that the bacteria had already reduced the nitrate to nitrite. There was nothing left for the reagent to reduce. But in C, so same situation as in B here, we've got a negative result, or we've got no color change, but we don't know if that's due to further reduction to nitrogen gas, or if that it did not reduce. So we add our zinc dust, then we get a yellow color, a red color, which means that the nitrite was still present in the sample, so the bacteria had not reduced it, the zinc dust did. Okay, and then here's just a different kind of drawing that goes through each one of those steps. So you can look through that. Okay, next is oxidase, which we've already talked about this test, but it detects the production of cytochrome oxidase, which this is a very helpful test in differentiating between the Enterobacteriaceae and which are all oxidase negative fermenters from the fermenters. So a purple result is positive, which means that the organism does produce cytochrome oxidase, so it's referred to as a fermenter, versus no color change, which is a negative result, which means the organism does not produce cytochrome oxidase, so the organism would be referred to as a non-fermenter. And there's that same picture that you've seen previously of those results. Okay, the next test is ONPG. So organisms that are delayed lactose fermenter. That just means that they will utilize lactose, but it just takes them a little bit longer to do that. But because of that, they can appear initially as non-fermenting colonies on your isolation media. So the ONPG test determines whether the organism is a delayed lactose fermenter or if they are a true non-lactose fermenter. So it tests for the enzyme beta-galactosidase. We do this by making a heavy suspension in saline with the organism, and then we add an ONPG disc. So that suspension is incubated at 35 degrees Celsius. Positive results can be seen within 6 hours. Negative results, your broth will remain colorless. A positive result is yellow. So that means beta-galactosidase is present, so it is a delayed lactose fermenter. Bacteria that produce a yellow pigment should not be tested because that can result in a false positive. So your lactose fermenters produce beta-galactosidase and permease, both of those enzymes. Your delayed lactose fermenters produce only beta-galactosidase, so they're going to appear as non-fermenting colonies on the media. And then your non-lactose fermenters don't produce either enzyme. So there's an example of those reactions. Okay, next test is the PAD, phenylalanine deaminase, which determines whether an organism possesses the enzyme that deaminates phenylalanine to phenylpyruvic acid. So we're going to inoculate the slant, incubate, add our 10% ferric chloride, which is our reagent. Positive result is green on the slant. Negative result is no color change. This test is especially helpful to differentiate Proteus, Morganella, and Providentia species, which are PAD positive, from the rest of the organisms in the Enterobacteriaceae family. And there's a positive and negative result for that one. Next test is the MUG test, which detects the hydrolysis of a substrate to a fluorescent compound. So a positive or a result, what we're looking for in this test is a fluorescent blue color under long wave UV light. So this one is used for the presumptive identification of your pathogenic strains of E. coli. So that's your virotoxin producing your O157H7, and to differentiate it from the other Enterobacteriaceae. And this one is a little bit counterintuitive. So if the result is negative, meaning that no fluorescence is produced, this means that it's positive for our pathogenic E. coli, the O157H7. If it's positive for fluorescence, that means it's one of these other strains of non-pathogenic E. coli. Okay, and here's a picture of that. So we have no fluorescence here, but the negative reaction is indicative of the pathogenic organism in this one. So negative is indicative of E. coli O157H7, whereas positive fluorescence is just one of the other strains of E. coli. Okay, a few final slides here. So rapid identification, and we mentioned this previously, I think in Unit 2. So there are several automated systems available, most of which use turbidity, colorimetry, or fluorescence technology to determine the identification of the organism. And then the fully automated systems incubate and read those reactions. Computer software interprets those reactions, and that's what it uses to determine the identification of the organism and sensitivities. And I showed you the picture before of the microscan. So this is one automated system. Vitec is another. And whereas microscan is considered an open system because there's nothing that contains the reagents and the inoculum in that panel. Vitec, and I don't have a picture of it here, but it's like the size of a credit card, and everything is contained inside of that card. We've also got, and some of these systems can give you an identification, in 4 to 6 hours, which for microbiology is pretty fast. We've got these rapid cartridge systems, and this is what I was trying to tell you about in Unit 2 when we were talking about the Neisseria. So it's kind of the same concept as those panels that are used for the microscan or the Vitec. We've got all of these little wells here that we inoculate with a broth that we've made from our organism. We have to incubate it for a few hours, and we come back and we're reading all of those color reactions that help us to get our identification. And then also the microscan and the Vitec systems provide us with drug sensitivities. Okay, a few little review slides here. Which of the following is not a general characteristic of the Enterobacteriaceae? They are not spore formers. Okay, the TSI test contains which sugars? Glucose, lactose, and sucrose. Which test detects ammonia as an end product? The urease test, because urease hydrolyzes urea to form ammonia. Delayed lactose fermenters produce which enzyme? Beta-galactosidase only. Okay, so that is the overview of the family of bacteria known as the Enterobacteriaceae, and common testing that is performed on those organisms. That's a lot of information. I know that, so that's why this last little bit has been going to be broken down into two different sections, but the next video we will actually be talking about the organisms that are in that family known as the Enterobacteriaceae.